the recording. Share my screen. All right, you should be seeing my screen now. Okay, today is February 10th, and we are supposed to be starting chapter 6. We're a little behind schedule. For the lab today, there is no lab. You're supposed to be working on your unknown work. I'll be logging in for about 15 minutes to answer any questions. Any questions about what we're doing? All right. So last time we talked briefly about anaerobic respiration, saying that it's similar to aerobic respiration. However, instead of the final electron acceptor being oxygen, it's some other molecule other than oxygen. And I forgot to put this in. And the total number of ATP is less than is generated in aerobic respiration. The number is between 2 and 36, depending on which form of anaerobic respiration it is. Any questions about any of that? Unplug that. Plug in my headphone. I don't have enough ports to have everything plugged in, so go back. All right, this slide is showing you a summary of aerobic respiration and fermentation. Can you guys hear me? I'm talking through my headphone now.
Yes.
Okay, good. So we're going to talk about fermentation. And fermentation does generate ATP, but it does not generate much ATP. It releases energy from the oxidation of organic molecule, mainly glucose. And this generation of ATP is always generated by glycolysis. Glycolysis does proceed, or is a part of aerobic respiration. It is also a part of fermentation. As previously discussed, we generate 2 pyruvic acid molecules, 2 net ATP molecules, NADH and hydrogen, from oxidizing glucose. You can also use other carbohydrates, amino acids, organic acids like fatty acids, and nucleotides. And I think we're going to talk about a little bit of that in just a little bit, but we haven't really talked much about that. But glycolysis prefers to use glucose, but it can use other molecules. Any question about any of that? So in fermentation, what we do is we run glycolysis, and then we have 1 or 2 fermentation steps after glycolysis. So pyruvate at the end of glycolysis is converted to the fermentation molecule, and it can vary on which fermentation reaction we're doing, but it can be lactate, it can be alcohol, it can be a few other molecules as well. Fermentation does not require oxygen, so it can happen when oxygen is present or when oxygen is absent. It does not use the Krebs cycle or the electron transport chain. It only uses glycolysis and then 1 or 2 fermentation steps after glycolysis. The electrons do move from glucose, and then we generate NADH, and the electrons from glucose are going there. The electrons are flowing from glucose, flow to an organic molecule as the final electron acceptor. And then the pyruvate is converted into the end product of fermentation. The conversion of pyruvate to the end product of fermentation uses the NADH that is used up by glycolysis or made by glycolysis. And then that NADH is recycled into NAD+. Why would the cell use up NADH to undergo fermentation if the cell could store that NADH and then put it through aerobic respiration? Because you generate more ATP with aerobic respiration than you do with fermentation. Anybody have a guess? You're actually seeing it right here. And that is you have to regenerate the NADH and regenerate NAD plus from NADH so that glycolysis can occur. If you have no NAD plus, glycolysis cannot occur. So NAD plus is required for glycolysis to run. And so the cell in fermentation regenerates the NAD plus for fermentation and glycolysis to occur. Without NAD plus, the cell cannot generate any ATP in glycolysis. So the cell has to recycle the NADH in order for glycolysis to occur. And yes, fermentation does not generate much ATP, only 2 ATP from glycolysis. But if there's no oxygen around, like your muscle cells are trying to work, they will fall back on fermentation to generate some ATP because the muscles don't have any oxygen, so they cannot perform aerobic respiration. And that's true for bacteria too. If they're falling back on fermentation, they can generate a little bit of ATP too in the absence of oxygen. But once oxygen becomes available, your muscle cell as well as the bacteria cell can then stop fermentation and engage in fermentation. Stop fermentation and engage in aerobic respiration. And when you do that, you generate much more ATP than fermentation is able to produce. There's 2 fermentation reactions we're going to talk about. There's about 5 or 6 total fermentation reactions. In lactic acid fermentation, pyruvate is generated by glycolysis, and we do generate 2 ATP. And then the NADH is converted into NAD plus in 2 fermentation steps. And we generate from the pyruvate lactic acid This is an ingredient that's actually important to prevent food spoilage. We find lactic acid in sauerkraut, in yogurt, in many fermented foods like most cheese, like cheddar cheese, and chocolate. Another fermentation reaction is alcoholic fermentation, which, oh, I'm showing you the wrong one. I got to blow that up. Lactic acid fermentation is this. It happens in only one step. Alcoholic fermentation is this one, and it happens in two steps, where pyruvic acid is converted into ethanol in two steps. Ethanol is only a 2-carbon molecule. Pyruvic acid is a 3-carbon molecule. And so we generate CO2 in alcoholic fermentation. And that's how you get the fizzling in beer, the CO2 being generated. In wine, we do not generate, well, we do generate CO2, but the CO2 is vented to the atmosphere and comes off of the wine. If you were to bottle that bottle of wine and then keep the CO2 in the wine, we call that champagne, and that is bubbly wine. But normally wine is made in either an open casket or an open barrel, and then the CO2 is just vented to the atmosphere. Any questions about alcoholic fermentation and lactic acid fermentation? Alcoholic fermentation is only done by yeast, Saccharomyces cerevisiae, making ethanol. There's a couple of bacteria that make lactic acid fermentation. You don't really need to know the species, but this is how we make sourdough bread and, well, cheddar cheese, and most of the fermented food that we have. Follows lactic acid fermentation. There is one exception, and that's Swiss cheese. Uses propionic acid fermentation, and that causes CO2 to be generated, and the CO2 makes the holes that we see in Swiss cheese. There's also butyric acid fermentation and mixed acid fermentation, and another mixed acid fermentation, which Enterobacter makes. And so these are some of the fermentation reactions. Like I said, you really only need to know about lactic acid fermentation and ethanolic fermentation. But the point is there's more than one fermentation reaction, and it generates different end products. Fermentation is used commercially to make different products. Like ethanol fermentation is used to make beer and wine, and we use that to make the alcohol that you can find in your gasoline as well, fuel. Acetic acid fermentation is used to make vinegar, and that actually works on ethanol to make the vinegar. So that first uses ethanolic fermentation, whether it's up here to make the wine, and then you use the wine to make the vinegar. Lactic acid fermentation, as I stated, does a lot of products: cheese, yogurt, rye bread, sauerkraut, sausage. It's fermented. And most fermentation food is with lactic acid fermentation. Industrial uses, the making of acetone and butanol, glycerol, methane, which is also a fuel product. All right, any question about any of that? If not, this is comparing the different forms of respiration to fermentation, which some people call fermentation a form of anaerobic respiration. I do not, and your textbook does not, because, well, fermentation is a fermentation reaction, and I consider anaerobic respiration to be similar to aerobic respiration. And it involves glycolysis, the Krebs cycle, and the electron transport chain. The electron transport chain is just different. And instead of the electrons flowing to oxygen and aerobic respiration, in anaerobic respiration, the electrons flow to another molecule other than oxygen. And in fermentation, the electrons flow to an organic molecule. In the generation of ATP with aerobic respiration, we have substrate level phosphorylation in glycolysis, and then oxidative phosphorylation in the electron transport chain generating ATP. You have the two same in anaerobic respiration, but in fermentation, we only have substrate level generation of ATP. And then the generation of ATP molecules in aerobic respiration, we generate either 36 or 38 ATP molecules. In anaerobic respiration, it depends on which anaerobic respiration we use, but it's some number between 2 ATP molecules and then 36 ATP molecules. And then in fermentation, you only generate 2 ATP molecules. And all of these ATP molecules are made by glycolysis. All right. So we've talked about the catabolism of a glucose or some other carbohydrate, which is used as the food source for organisms to catabolize them, to catabolize this carbohydrate to generate ATP. But other molecules can be used. Like I said, other sugars can be used and they can be converted into glucose and then use aerobic respiration. You can use other carbohydrates like starch, break the starch down into glucose and then send that through ATP. You can use other large molecules like lipids and proteins. And there's a problem with digesting large molecules, even starch. And that's because they're too large to come into the cell. And what the cell does is the cell, oops, why did that blow up? Why do I have this over here? What the cell does is it produces enzymes to digest the large molecule outside the cell. And then that large molecule is broken down into smaller units, the monomers. And then the monomers are brought into the cell. So for starch, you break down the monomers into glucose. And then you bring the glucose into the cell by active transport. There's a transport protein which brings the glucose across the cell membrane. For lipids, the cell produces lipases and then secretes those enzymes extracellular digest the lipid outside the cell, making fatty acids and glycerol, and then bring the smaller molecules into the cell by active transport. For proteins, the cell will make proteases, digest the proteins outside the cell, forming amino acids, and then bring the amino acids into the cell by a transport protein. Any question about any of that?
I have sort of an off-topic question. I've
Uh-huh.
heard about like people reducing their carbohydrate intake and reducing glycolysis while increasing their fat and protein intake in an attempt to get the body to break down fats through the keto diet. I was wondering if there was any truth in that or if they were just like messing around.
When they do that, they generally convert the lipids and proteins into glucose and then send glucose through aerobic respiration in the normal way. That's the way all couch potatoes digest lipids and proteins. However, if you're an athlete, you can digest the lipids directly. However, the marathon runner, which they can digest the lipids directly, they take 45 minutes in their run before they digest the lipids. So a marathon runner has to be running for 45 minutes before they can directly digest the lipids, but they can directly digest the lipids. And I don't know how long it takes before you have to be in your run for a marathon runner to start digesting proteins, but they can also digest proteins. But most of us are couch potatoes, at least most of the time, in which case the lipids and proteins will be converted into glucose, and then glucose will go through aerobic respiration in the normal way.
Is it possible to like increase the rate of fats being converted into glucose by just reducing your carbohydrate intake?
Can you repeat that? I didn't hear all of that.
Is it possible to like increase the rate of fats being converted to glucose by reducing your carbohydrate intake?
Yeah, if you're not eating enough carbohydrates, you are going to digest lipids and proteins. You always digest the lipids and proteins. I mean, that always is happening anyways. But my point is, if you reduce the carbohydrates, you will be burning more lipids and proteins to make your ATP. And if you simply eat more food than you need to generate ATP, the food is still going to be digested and it will be stored down as fat by the body. So it is true that if you have a high or low carbohydrate diet, it takes a little bit more energy to digest the food. So I guess you can eat a little bit more because it takes more energy to digest the food. And so some of that energy has to be used up to digest the food. But in reality, you're not gaining very much. Okay. And there is a problem with having a high protein diet, and that is the amino group has to come off of each amino acid before that amino group can be converted into a carbohydrate and then used by aerobic respiration. And then this amine group that's removed from the amino group of an amino acid is then converted into urea and has to be peed out. And that takes energy to do that as well. The monomers from lipids and proteins interaerobic respiration or either in glycolysis or the Krebs cycle, and then they're digested in the normal way. Let's talk a little bit more about each of these. So for a carbohydrate, let me blow this up a little bit. Carbohydrates are very easy to digest. The large carbohydrates are simply converted into the simple sugars, and then the simple sugars are converted into glucose, and then glucose is sent through aerobic respiration in the normal way, which I'm not really showing it, all of it, but there we go. The Krebs cycle and electron transport chain, and then we make ATP in the normal way. Proteins are a little more complex, and the proteins are digested into amino acids. The amino acids are then brought into the cell. Depending on which amino acid it is, some amino acids are put into glycolysis, like around pyruvic acid, and then converted into pyruvic acid. Some amino acids are converted into acetyl coenzyme A and then enter the Krebs cycle. Other amino acids enter the Krebs cycle. And then the protein goes through aerobic respiration in the normal way. Okay, although you may be entering aerobic respiration lower down in the chain than you do for entering with glucose. For lipids, lipids are broken outside the cell by an enzyme into glycerol and fatty acid. The glycerol and the fatty acids are then brought into the cell. And this is true for both our cells and for bacteria cells. The fatty acids are converted into 2 carbon units. The 2 carbon units are converted into acetyl coenzyme A, and then acetyl coenzyme A enters the Krebs cycle in the normal fashion. The glycerol is converted into a carbohydrate of the glycolysis, and then the glycerol enters glycolysis the normal way. And then the end product is we go through aerobic respiration, generating ATP, regardless of what food molecule we started with. Why did I lose that? There we go. So regardless of what the food molecule is, it will be digested and enter the aerobic respiration. Any question about any of that?
Can you zoom out a little bit?
Is that better?
Yes, thank you.
Right. All right, the complete metabolic oxidation of glucose in aerobic respiration is the most efficient for generating ATP, but the cells can use other molecules to digest and digest the food molecule and then generate ATP. And then the ATP is used by the cell for its energy needs. And this includes active transport of molecules for the cell's movement or for your muscles to move. And the cell uses ATP to synthesize new cellular components. Like when we're building protein, we will use ATP to link together the amino acids to make the protein. And the cell uses that for all forms of anabolism. The cell uses ATP for most of its cellular processes. And the one that uses the most is, of course, The cell, when it's biosynthesizing larger molecules like polysaccharides, lipids, and proteins and nucleic acids, the four molecules of life. To make the larger biological molecule, the cell has to use ATP. Shrink that down a little bit. So we've talked about anabolic and catabolic pathways as being distinct. However, in the cell, even in our cells, they are not distinct. The two pathways have common intermediates, and so that an anabolic process and a catabolic process are actually joined together. And if the pathway can go both ways, we call it an amphibolic pathways. Sometimes it will be catabolic, but if we make the reaction go the other way, it will be anabolic. Even human cells have amphibolic pathways where the pathway functions in both ways. And human cells are not very efficient, or meaning that they have much fewer amphibolic pathways than, say, green plant cells or green bacteria cells. In fact, all bacteria have more amphibolic pathways than human cells do. But the point is that amphibolic pathways bridge the pathways in carbohydrate, lipid, protein, and nucleic acid metabolism. And as I stated, most of us couch potatoes, when we consume and digest proteins and lipids, we don't burn them directly. We will convert the protein and lipid. Let me see if I can blow this up. If I do it from the slideshow, that should do it. It blows it up a little bit. Not real good, but you can sort of see wherever there's two arrows, one going this way, one going that way, it means that in human cells, we can go both ways. And so the amino acid here can go this way, forming pyruvic acid, and then the pyruvic acid can go this way to form glucose. And that happens, of course, most carbohydrates will be broken down to a simple sugar and then they can be converted into glucose directly. But for proteins, for most of us couch potatoes, we don't burn proteins directly. We will convert them into glucose and then send glucose through aerobic respiration in the normal way. And the process for making glucose is actually quite complex for a protein. Like this amino acid enters late in the Krebs cycle, and we have to go all the way this way to get glucose. And the same thing for lipids. You break them down into glycerol and fatty acids. The fatty acids are converted into acetyl coenzyme A. If we directly burn the acetyl coenzyme A, we can go this way. But if we don't, we go this way and then convert the acetyl coenzyme A into glucose. And then glycerol is converted into this, and then we can go that way to make glucose. All right, any question about amphibolic pathways? The conversion of one molecule into another, generally making glucose, so that glucose can then go through aerobic respiration in the normal way. As I stated, a marathon runner has to be running for 45 minutes before they can directly burn lipids. Okay. Human cells are not very good at being amphibolic. This is not showing you that human cells can combine glycerol and fatty acids to make lipids, but that can be done. As you know, when we eat too much carbohydrates or even too much protein, those will be converted into glycerol and fatty acids, which will be stored by your body as a lipid, a fat, and then we'll put down fat. So even if you eat too high of a protein diet, you will still start storing, if you eat too much of that protein, you will store that down as lipids. Anything that's not burned will be stored as a lipid. And that's, I guess, not entirely true. Our body can store carbohydrate in our liver as glycogen. So some of the lipids could be converted into glycogen and then stored in the liver. But most of the excess food we eat will be stored as fat. Green plants are much better at engaging in amphibolic pathways. And as you know, green plants can generally, from CO2 and water and minerals that they take up by their roots, they can make any molecule that the green plant needs. We are not able to do that. Like there are essential fatty acids and essential amino acids. One essential amino acid is lysine, and this is an amino acid we have to get from our diet. If we don't get it from our diet, we would become unhealthy because we cannot make lysine, and we need lysine, and eventually we would die because we just can't make that amino acid. So we have to get it from our diet. And it's usually pretty easy to get. All you have to do is eat protein and you're going to get lysine. All right, any questions about carbohydrate metabolism? If not, let's mention that ATP can also be made by catabolism, but in some organisms, they can make ATP by anabolism. All of the photosynthetic organisms can make ATP by photosynthesis. Photosynthesis is the conversion of light energy into chemical energy. The chemical energy is used usually to convert CO2 into glucose, and that we call carbon fixation. Green plants can do this, but cyanobacteria, algae, and other photosynthetic bacteria can also do this. And this is a mechanism that other organisms need to get organic compounds. All animals and microbes that feed on the photosynthetic organisms need the photosynthetic organisms to make glucose. Without it, we could not survive. Even if you were to eat the cow has to eat the grass. And so someplace along the line, we need the photosynthetic organisms to, well, make the food that we eat. Like I said, even if you eat the cow has to eat the grass. And there is one exception, and that is there can be chemosynthetic organisms which can convert chemicals in one, or the electrons in one chemical, and use it to fix glucose. So chemosynthetic organisms and photosynthetic organisms are can make ATP by anabolism. We're not going to talk about chemosynthetic organisms, but I will briefly talk about photosynthetic organisms. Only photosynthetic organisms can engage in photosynthesis. They contain light-trapping pigments. One of them, you know, is chlorophyll. The light releases the electrons from chlorophyll. These electrons then begin moving, and the electrons can be used to make ATP. Light energy is converted to chemical energy by electron transport chain. We have carrier molecules. I'll discuss that in just a minute. So photosynthetic organisms do make ATP and or NADPH. Usually ATP and NADPH are used up by the photosynthetic organism to make glucose from CO2 and water. And so you do know one photosynthetic reaction, and that's the non-cyclic photosynthetic reaction, but there's actually two photosynthetic reactions, the cyclic and the non-cyclic. In the non-cyclic photosynthesis, this is where carbon dioxide and water in the presence of light are converted into glucose. And this is the photosynthetic reaction you've probably seen previously. This, oops, I didn't mean to change that. This can generate oxygen. You should know there's another photosynthetic reaction. You don't need to know it, but it doesn't generate oxygen. So this is oxygenic photogenesis. And this is the one you've probably seen. Water is generated to generate glucose and oxygen. There is another photosynthetic reaction you've probably never been told. And in this cyclic photosynthesis, light energy is used to make ATP. So the only thing that happens in cyclic photosynthesis is ADP plus phosphate generates ATP. All photosynthetic organisms can engage in both cyclic and non-cyclic photosynthesis. If the cell needs ATP, like a green plant cell, it will engage in cyclic photosynthesis. But if the cell wants glucose, it will engage in non-cyclic photosynthesis. Let's talk a little bit more about cyclic photosynthesis. Light strikes the photosystem, and there's chlorophyll in the photosystem. The light causes two electrons in chlorophyll to start moving. The electrons move to this protein in a membrane, to this protein in the membrane, to this protein in the membrane, and then the electrons move back to chlorophyll. And that's why it's called cyclic photosynthesis, because the electrons move in a cycle and they come back to where they started from. Cyclic photophosphorylation. But as the electrons are moving, when they come through this protein, they give the protein the energy it needs to pump hydrogen from this side of the membrane to that side of the membrane. And the hydrogen builds up on this side of the membrane, creating a potential energy difference where the hydrogen wants to come over to this side of the membrane. And the hydrogen can only move through this channel protein, which is ATP synthase. And when the hydrogen move from this side of the membrane to that side of the membrane, They give this protein the energy it needs to make ATP. This should sound familiar to you because it's very similar to the electron transport chain in aerobic respiration. Electrons are moving. Electrons give the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. The hydrogen ions then are used to make ATP. Very similar to aerobic respiration, or the electron transport chain of aerobic respiration. The difference is that the electrons are moving because of light, and in aerobic respiration, the electrons come from glucose. Another difference is that aerobic respiration in eukaryote cells happens on the inner mitochondrial membrane. Photosynthesis happens in the inner mitochondria, not the inner mitochondria, the inner membrane of the chloroplast. In bacteria, which do not have chloroplasts, this happens on the cell membrane. So any question about that? And then these electrons are moved, or the hydrogen ions are moved from inside the cell to the cell wall, creating the hydrogen gradient. All right, so let's talk a little bit about ATP production. The energy source can vary. There can be different electron donors. In carbohydrate metabolism, the molecule can be glucose, where the electrons come from, or it can be from some other molecule, like another food molecule we're digesting. In chemosynthetic bacteria, it can be another chemical like sulfur or ammonia. In photosynthetic organisms, the electrons start moving from light hitting the chloroplast, causing the electrons to move. So the point is the electron donor can vary. The electrons are then carried by a carrier molecule. Along the way, ATP is generated. Let me shrink that down a little. And then the final electron acceptor can vary. In aerobic respiration, the final electron acceptor is oxygen. In anaerobic respiration, the final electron acceptor is a molecule other than oxygen. In fermentation, it's an organic compound that accepts the final electron acceptor. And what's the final electron acceptor in cyclic photosynthesis? That's not shown here. But in cyclic photosynthesis, where does the electrons flow to? Nobody remembers? Does this help you out? Showing you right here. All right, nobody's going to comment, so we're just going to move forward. Any questions about the generation of ATP? If not, let's move on to chapter 6. All right. This slide is showing you my typical slide, the major goals of the lesson, microbial growth, and the rough outline. Know the terms, understand the various requirements the cells have for growth, understand the 4 phases of growth, and be able to compare the different methods for measuring microbial growth. When we're talking about microbial growth, you should realize that we're referring to an increase in the number of cells, not an increase in the cell size. Got a little video for that. The cells have various requirements for microbial growth. There are physical requirements and chemical requirements. This doesn't look like this is going to work. Let me try it in another browser. Yeah, what is that working? Let me try this one. I don't understand what this even is. Is that the same link I've got here? That was not converting. All right. I'm not sure why that link isn't working. I'll have to look into that. It's going to show you cells growing. There are physical requirements for microbial growth, including temperature, pH, osmotic pressure, salinity. There are also chemical requirements for cells to grow. All cells have to have a source of carbon, a source of nitrogen, a source of sulfur, a source of phosphorus, a source of trace elements. Cells have different oxygen requirement, and they may have organic growth factors that are required to grow. Like our cells require vitamin C, or else our cells are not going to be able to stay healthy and grow. Any question about any of that? Each species has a temperature requirement. Each species has a minimum temperature that it'll grow at, a maximum temperature it'll grow at, and an optimum temperature it will grow at. When we're talking about microbes, we can divide them into psychrophiles. These are cells that have an optimal growth rate around 10 degrees Celsius. This is refrigeration temperatures, psychrophiles. They like growing at very low temperature. And there are bacteria that can grow in your refrigerator, the psychrophiles, and they can spoil your food. Why most food in the refrigerator should be thrown away after a week, because the psychrophiles will grow and can spoil the food. The psychotrophs grow above that. They grow around room temperature. The mesophiles grow around 37 degrees Celsius. That's 98.6 degrees Fahrenheit. All of the human pathogens are mesophiles. So this is where all of the human pathogens are. The thermophiles grow best around 60 degrees, 65 degrees Celsius. And if you see the rotisserie going around at Safeway or some other store, and you look at it and you say, gosh, that chicken looks kind of dry, the thermophiles can grow at that temperature. The hypothermophiles grow best at temperatures above 90 degrees Celsius. 90 degrees to 100 degrees Celsius, around 95 degrees. Remember, 100 degrees Celsius is the temperature of boiling water. Now you should note that the ranges and even the optimal temperature for a specific organism is not ironclad, meaning The mesophile, not all mesophiles have an optimal temperature at 37 degrees Celsius. For example, Micrococcus luteus has an optimum growth rate around 32 degrees Celsius. We still call it a mesophile. Micrococcus luteus grows on your human skin, and that temperature is a little cooler than the rest of your body. And that's why Micrococcus luteus grows best around 32 to 35 degrees Celsius. Any question about any of that? The range also is not ironclad. Different species will grow at a different minimal temperature and a different maximal temperature. So it really depends on the species, what its minimum, its maximum, and its optimal temperature is. But this is giving you the approximate optimum and the range, meaning the optimum for all mesophiles is close to 37 degrees. Any question about that? All right. Let's briefly talk about food spoilage. Roll that up a little bit. Microbes grow very well between 60 and 130 degrees Fahrenheit. That would be something like 15 degrees Celsius to 52 degrees Celsius. And in this range is the danger range. Any food that is left out for more than 4 hours at this temperature should be thrown away because, or refrigerated, because the bacteria can grow rapidly at these temperatures and then produce toxins which can spoil the food. Now, above that and below that, from temperature 40 degrees to 60 degrees Fahrenheit, and from 130 to 45 degrees, bacteria can grow, but their growth is slow. At lower temperatures like 0 degrees Celsius, that's 32 degrees Fahrenheit, the bacteria will not grow because the food is frozen. And then at very high temperatures, the bacteria that cause spoilage of food cannot grow. Hypothermophiles can grow, but for some reason, those don't seem to spoil food. I'll talk briefly about that. So food that takes too long to cool, like you put it in a very large batch and then add it to your refrigerator, the food cools too slowly. The bacteria then have several hours to grow, and the growth is good enough for the bacteria to produce toxins. That food should not be eaten. You want the food to be in small batches to cool quickly, and that way the bacteria cannot grow. And let me see how many hours that is. Probably you want it to go through the danger zone in an hour or less. All right. Bacteria have a pH requirement. Most bacteria grow best between pH 6.5 and 7.5. However, there are bacteria that do grow in different environments, like an acid environment. Acids generally, once they build up, inhibit the growth of the bacteria. We minimize the acids growing, building up in the solutions in our microbiology lab by adding pH buffers to the culture media. Molds and yeast grow best in slightly acid conditions between pH 5 and 6. But acetophiles can grow in acid environments, like volcanic environments between pH 0 and pH 3. As you know, the stomach is between pH 1 and pH 2, and acetophiles can grow in your stomach. So acetophiles are bacteria that love acid environments. There are some bacteria that grow under only high osmotic pressure. Most microbes require water in order to grow. And if you don't have enough water around and you have salt or sugars around, the bacteria cannot grow. Most microbes grow well in osmotic pressure below 1 to 2%. Above that, they cannot grow, or at least grow slower. Excuse me. But some microbes grow in high osmotic pressures. High osmotic pressures removes the water from organisms that do not live in high osmotic pressure. So an isotonic solution, the water stays in the cell. Under high osmotic pressure, the water leaves the cell, the cytoplasm shrinks, that leads to plasmolysis, which will inhibit the cell and may kill the cell. High salt and high sugar concentrations can do this, and we can use this to help preserve the food, like salted meat, like salted fish, and molasses. Why the molasses is preserved? Because if the bacteria fall in the molasses, the water leaves the cell, and then the cell cannot grow. Halophiles, though, can grow in salt area. Halophile literally means salt lover. There are obligate halophiles, and they require a high salt concentration to grow. High salt concentration is above 7% sodium chloride. The highest halophiles can grow at 30% sodium chloride, and you can find 30% sodium chloride in the Dead Sea between Palestine and Jordan. There are also facultative halophiles. They do not require high salt concentrations to grow, but they can tolerate salt above 2%. Some facultative halophiles can grow at 15% sodium chloride. An example of a facultative halophile is Staphylococcus aureus. It can grow on your skin, which can get up to 20% sodium chloride. But Staphylococcus aureus can grow in a normal TSA plate, so they do not require high salt in order to grow. In a hypotonic solution, this is a low osmotic pressure solution, meaning there's lots of water around, very little salt. Water enters the cell by osmosis. If the cell does not have a functional cell wall, the cell may lyse. And that's actually how Penicillin G and the other members of the penicillin family lyse the cell. They interfere with the cell wall. And then the cell can lyse as water enters the cell. And the cell wall doesn't protect the cytoplasm, and it protrudes out of the cell wall, and then the cytoplasm will burst. Cells have a carbon requirement. Carbon is typically half of the weight of a dried cell, meaning if you dry and remove the water from cells, half of the weight of the cell is carbon. Carbon is used by cell to make structural organic molecules. Carbon is also an energy source, like glucose can be used to make ATP. Heterotrophs use an organic carbon source, such as glucose. Autotrophs, though, can use carbon dioxide, an inorganic carbon source. Autotrophs get their carbon from carbon dioxide. Nitrogen is another chemical requirement that cells need. Nitrogen is actually 14% of a cell's dry weight. You find nitrogen in amino acid, proteins, DNA, RNA, and ATP. Human cells usually obtain decomposition of nitrogen from decomposing proteins, meaning we digest proteins. This is true for many bacteria as well, but bacteria can obtain their nitrogen from other sources other than proteins. Many bacteria can use ammonia or ammonium to obtain their nitrogen. Some bacteria can use nitrate as a nitrogen source. A few bacteria can even use nitrogen gas in the atmosphere, and they fix nitrogen. They pull the nitrogen out of the atmosphere, fixing it so that other organisms can use that nitrogen. Sulfur is also a requirement of cells. Most organisms obtain their sulfur from either amino acids or proteins, and that's the way we get our sulfur. Sulfur is required in thymine and biotin and for DNA and RNA synthesis. Like I said, we get our sulfur from decomposing proteins. Bacteria can get their sulfur from decomposing protein, but many bacteria can get their sulfur from sulfate or from hydrogen sulfide gas. We cannot. Hydrogen sulfide gas is toxic to us. Green plants can get it from sulfate, but we cannot. Phosphorus, another chemical requirement. Phosphorus is in DNA, RNA, ATP, and in the cell membranes. Phospholipids have phosphorus in it. Phosphorus is usually in phosphate, shown here. It's an important source of phosphorus for green plants. Phosphate is a nutrient. We get most of our phosphorus from the food we eat, like milk and meat have phosphorus in it. Trace elements are inorganic elements required by microbes in order to survive. They require these trace elements in small amounts. Usually trace elements are just picked up from the environment. Trace elements would be something like copper, magnesium, iron. Usually we get enough of these in the food we eat and in the liquids we drink. This is true for bacteria too. All of them have trace elements, and usually they get the trace elements directly from their environment. the atom just diffuses across the cell membrane. Occasionally, we may live in an environment where we need to supplement that with trace elements. For those of you who live in Western Washington or Western Oregon, the rain has washed away iodine from our environment. And so if you were to only be eating local food, meaning food grown in Western Oregon or Western Washington, you would not get enough iodine. And so we have to eat food from other locations to get iodine, or you can supplement iodine in your diet. And iodine is added like in table salt. Trace elements are often required as a coenzyme. As a cofactor for an enzyme. And examples are iron, copper, and zinc. Organic growth factors are biological molecules that the organism needs in order to survive. So the organism cannot synthesize an organic growth factor. Examples of a human organic growth factor are various vitamins like vitamin C, certain essential amino acids. That's why we call it an essential amino acid. I mentioned lysine as an example. Essential fatty acids. Some organisms require purines and pyrimidines. We get these in our diet. So if we have an organic growth factor, we generally call it something like an essential amino acid or a vitamin, like vitamin C. We cannot make those elements. Green plants and photosynthetic bacteria can make all of those organisms, but some bacteria may have an organic growth factor. Most bacteria can make vitamin C, and mice can make vitamin C. We still call it vitamin C, even though they can make that. So the name vitamin is actually a misnomer, vitamin C, because that's something we have to eat. But mice and most bacteria cannot. If they do requirement, they must get it from their environment. Oxygen is a chemical that is required by some organisms. However, oxygen is, in a sense, a toxic substance. Even for those of us who must have oxygen, oxygen is toxic. There are 4 toxic forms of oxygen. Singlet oxygen, where oxygen is boosted to a higher energy state. This oxygen is highly reactive. It's actually found in phagocytic cells, like phagocytic white blood cells. And then the white blood cell releases singlet oxygen on something the cell is trying to kill. So if something has invaded the body and the white blood cell tries to kill it off, it can release singlet oxygen to try and kill whatever has invaded the body. There's also superoxide free radicals. This is oxygen with an extra electron. So that extra electron gives the oxygen a negative charge. So that symbol refers to an unpaired electron. It's only found in free radicals. These molecules are highly unstable. Whenever they bump into another molecule, they will grab an electron from that molecule, meaning oxygen with the free electron wants to pull another electron or give up that electron. Superoxide free radicals are made in aerobic respiration. And usually the superoxide free radical steals an electron from another molecule it bumps into. And then that molecule that loses the electron bumps into another molecule, which can then steal an electron. These molecules need to be neutralized. And we have superoxide dismutase, or SOD, which can take these free radicals and then convert them into catalase. Catalase is still a problem for the cell, but on the next slide, I'm showing you that catalase, hydrogen peroxide is a problem for the cell, but it can be neutralized by the enzyme catalase. All right, any question about the toxic forms of oxygen as we've discussed so far? If not, I'm going to stop here and I will see you in the lab at 6:30 if you have a question. If you don't have a question, you do not need to show up for the lab today. Make sure today is Thursday. Yep. All right. If there's no questions, I'm going to end it here. And I'll see you later.